Usually when we talk about a medical diagnosis or just biology in general, there's this expectation of precision.
Right, like it's a math equation or something.
Exactly. It feels like engineering. Like you break your arm, the x-ray shows that jagged white line, and the doctor just points at the screen and says, well, there it is.
Yeah, very cut and dry.
But then you step into the world of neurodevelopment and reproductive biology, and suddenly that x-ray machine feels like it's just broken.
Oh, completely. It gets so murky.
Right. And we hear terms thrown around in popular culture all the time, like, mom brain.
Oh, yeah, the classic punchline.
Exactly. It's almost always a punchline. It's just this convenient way to describe, I don't know, forgetting where you put your car keys or inexplicably putting the milk in the pantry.
Yeah, I mean, it is entirely framed as a deficit, like a temporary, exhausting glitch in the system.
A glitch, yes. But what if we've had it completely backwards this whole time? What if mom brain isn't a deficit at all, but rather a radical, lifelong biological upgrade?
A literal structural rewiring.
Exactly. A rewiring of the hardware that lasts for decades. So for you listening today, we are going deep into the exact molecular mechanisms of matrescence.
Which is the physical, emotional, and biological transition to motherhood.
Right. And the source material we have to guide us through this deep dive today is just absolutely incredible.
It really is. We are looking at this fascinating 2026 paper published in the journal Nature. It's titled Dopamine Drives Persistent Remodeling of the Maternal Brain.
And our mission for this deep dive is to uncover the precise physical mechanisms that reshape a mother's brain. And, how those changes don't just fade away after the kids grow up, but actually persist for the rest of her life.
Yeah, and to really understand the magnitude of this, it is so important to distinguish between acute changes and long-term changes.
Right, because usually we just hear about the pregnancy hormones.
Exactly. Historically, science has focused heavily on the acute changes during pregnancy. The immediate hormonal floods of oxytocin and estrogen that trigger parenting behavior right around birth.
The bonding stuff.
Yeah, but this research looks at something called reproductive experience, or RE.
Okay, let's define that. What exactly is RE in this context?
So in these mouse models, RE is the composite experience of pregnancy, birth, lactation, and actually caring for the pups over time.
Which is really important because it mirrors human parity, right?
Yes. Parity just means having carried a pregnancy to term. The researchers wanted to know what the brain looks like long after that initial hormonal storm has entirely passed.
Okay, so let's unpack the timeline here because I think it's the key to the whole mystery. We are talking about mapping the brain to find out exactly where motherhood lives, structurally.
Right.
So to do this, the researchers set up this fascinating experiment. They took two groups of mice, virgin females, which they call nulliparous, and mothers who had been through this full reproductive experience.
The catch is they didn't look at their brains immediately after birth.
Right. They waited until 4 weeks after the pups were completely weaned.
And that timeline is absolutely crucial to the integrity of these findings. Because by 49 days postpartum in a mouse, which corresponds to 4 weeks post-weaning, their circulating hormones have entirely returned to baseline virgin levels.
Okay, so no extra estrogen or oxytocin floating around to mess with the data.
Exactly. So any differences found in the brain at this point cannot be blamed on hormones. If there are changes, they are permanent structural or functional modifications to the brain itself.
Wow. So the researchers cast a wide net, profiling 11 different brain regions known to be involved in maternal behavior.
They were searching for DEGs, or differentially expressed genes.
Right, basically looking for which parts of the brain had the most genes permanently turned up or turned down. And the winner was incredibly surprising to me.
I know, it wasn't what you'd expect.
It wasn't the amygdala, our emotion center, and it wasn't the hypothalamus, the hormone control room.
No. The region with the most robust permanent transcriptional remodeling was the dorsal hippocampal formation, or DHF.
Which is such a fascinating twist, because the dorsal hippocampal formation is completely essential for our internal GPS.
Right. It is primarily responsible for spatial cognition, sensory integration, and detecting novelty in the environment.
And that is exactly where I get stuck. The hippocampus is universally associated with memory and navigation.
Yeah.
But I've always thought of maternal instinct as this deep emotional bond. Why is the navigation center the VIP region for motherhood rather than the emotional centers? It just feels so counterintuitive.
It does feel counterintuitive, well, at least until you look at it through a cold, hard lens of evolutionary survival.
Okay, tell me more.
A mother doesn't just need to bond with her offspring. I mean, she needs to keep them alive.
Right.
And to do that, she needs a massively upgraded internal map. Think about the cognitive load. She has to leave the nest, navigate incredibly complex environments to forage for limited resources, simultaneously detect subtle threats like predators.
All while keeping track of where she left her babies.
Exactly. And then successfully find her way back to the exact location of her highly vulnerable pups. The brain is effectively upgrading her spatial and sensory processing power to handle the massive processing demands of keeping multiple beings alive in a dangerous world.
Wow, okay, that makes so much sense.
Yeah,
It is literally a biological upgrade to her environmental radar.
it really is.
So just to do a quick recap for you listening, because we want to make sure these concepts really stick. Basically, matrescence causes lasting gene expression changes long after hormones fade.
Right.
And the DHF, our brain's GPS, undergoes the most robust transcriptional remodeling. So a question for you to think about. Think about a major life transition you've had. Did it feel like your brain physically rewired how you navigate the world? Because this shows it literally does.
Yeah, and that physical rewiring is exactly what the researchers wanted to visualize next. Because once they pinpointed the DHF as the site of the most intense remodeling, they needed to zoom in.
They needed to know what was actually happening to the specific cells inside.
Exactly.
And here's where it gets really interesting and honestly a little scary at first glance. When they looked closely at these highly sensitive brain regions, they found that the expression of general neuronal markers like the basic signs of overall brain cell activity actually went down.
Yeah, they decreased.
Let's pause there, because when I first read that, my immediate thought was, wait, does motherhood destroy brain cells?
Yeah.
Because if it does, that just validates every terrible joke about mom brain being a cognitive deficit.
I completely understand why it sounds alarming, but it is not destruction at all. What we are looking at is a highly specific, microscopic retuning of the neural circuits.
Okay, so how do they prove that it wasn't just, brain damage?
To prove this wasn't just broad cellular loss, the researchers utilized an incredibly advanced technique called single nuclei RNA sequencing, or snRNA-seq.
Wait, single nuclei RNA sequencing? I know RNA sequencing generally looks at the genetic instructions active in a piece of tissue, but pulling the nuclei out of single individual cells, how do you even separate that without getting a mishmash of all the surrounding tissue and missing the subtle details?
It's a great question because the precision really is mind-boggling. So imagine you have a massive bowl of mixed fruit. Strawberries, blueberries, raspberries, just all mashed together.
Okay, I'm picturing it.
If you just blend it up, you get a smoothie. You know generally what's in it, but you lose the specifics of each fruit. That's traditional sequencing.
Got it.
Single nuclei RNA sequencing allows scientists to isolate the exact recipe inside a single raspberry without the juice of the blueberries contaminating the data.
Oh, wow.
Yeah, they isolate the nucleus of individual cells and read the exact RNA transcripts inside just that one entity. And out of over 100,000 individual nuclei they sequenced, they found that the genetic changes weren't happening everywhere uniformly.
Where were they happening?
They were highly concentrated in a very specific cluster of cells called GABAergic neurons.
GABAergic neurons. Okay, so those are the cells that produce GABA, which is the brain's primary inhibitory neurotransmitter, right?
Yes.
They act like the brakes on a car, calming the nervous system down and keeping things regulated.
Exactly. And the changes were even more specific than just GABAergic neurons in general. They were happening in the GABAergic neurons that express two very particular receptors, dopamine receptor D1, known as DRD1, and dopamine receptor D2, or DRD2.
Okay, so dopamine receptors.
Right. And the single-cell RNA sequencing revealed that in maternal brains, the expression of these dopamine receptors was significantly dialed down.
Dialed down. Okay, my brain is snagging on this. Why would the maternal brain deliberately remove dopamine receptors? I mean, dopamine is our reward chemical. It's our motivation and learning driver. Shouldn't a mother's brain want more motivation?
It does, and that is the brilliant compensatory mechanism at play here. The researchers didn't just look at the receptors, they measured the actual baseline levels of the dopamine chemical itself in the maternal DHF, as well as in the nucleus accumbens, the brain's reward center.
And what did they find?
They discovered that baseline dopamine levels are actually permanently elevated in mothers.
Oh, I see. It's an adaptation. It's like, okay, imagine moving from a really gloomy, cloudy city to a place that is incredibly sunny all the time.
I like this.
The blazing sun represents that elevated, permanent baseline of dopamine. And because it is so much brighter all the time, you end up having to wear dark sunglasses or you close the blinds in your house a bit.
Yes.
Which would be the brain downregulating or removing those D1 and D2 receptors. You pull the blinds down just to comfortably adjust to this new permanent state of highlight.
That is a perfect analogy. The brain is relentlessly seeking homeostasis or balance. By maintaining higher baseline dopamine, the maternal brain is constantly primed for heightened motivation, vigilance, and sensory processing.
But to prevent the entire system from getting overstimulated and frantic, it pulls down the shades.
Exactly. It reduces the number of receptors. This delicate, lasting balance, high dopamine, lower receptors, is what gives the maternal brain its new, highly efficient set of capabilities.
It is such a profound shift in how we think about the chemistry of parenting. You always hear about the acute bonding hormones like oxytocin. But the idea that dopamine, the drive, motivation, and reward chemical, is actually the permanent architect of this change really stands out to me.
It reframes maternal instinct completely.
It really does. It moves it from just feeling love to driven, focused action. So, time for another quick recap for you listening. Reproductive experience changes specific GABAergic neurons. Maternal brains show a permanent increase in baseline dopamine, which leads to a compensatory drop in D1 and D2 receptors.
Yep, the sun and the blinds.
Exactly. So what stands out to you about dopamine being the key driver here rather than just the traditional bonding hormones? It's pretty wild to think about.
It is, but as the researchers discovered in the next phase of their study, this delicate rewiring process balancing the sun and the blinds is not guaranteed.
Oh, really?
No, it is incredibly fragile, particularly during a very specific window of time.
Right, which brings us to the disruption. The stress test. The researchers had to ask, what happens if this transition is interrupted?
And to figure this out, they utilized something called a maternal pup separation paradigm.
Which sounds awful.
It is stressful, yeah. Basically, between 10 and 20 days postpartum, they chronically stress the mother mice by repeatedly separating them from their pups for a few hours a day.
And why that specific window? 10 to 20 days.
Because that is absolutely critical. It's the developmental sweet spot when the brain is actively trying to lock in these new, upgraded dopamine circuits.
So they interrupted the download.
Exactly. And the outcome of introducing chronic stress during this time was biologically devastating. When they profiled the brains of the stressed mothers much later in life, those vital DHF adaptations had been severely disrupted.
So they didn't get the biological upgrade.
They did not. The stressed mothers failed to show that permanent baseline dopamine elevation. And because the dopamine never rose, because the sun never came out, to use your analogy, the brain never pulled the shades down.
It never downregulated the receptors.
Right. The entire transcriptomic remodeling process, the rewriting of the brain's GPS, was halted in its tracks.
Man, this part of the paper just hit me like a ton of bricks. Because of the real-world weight it carries, we talk about postpartum support for new parents all the time, right?
Yeah.
But usually it's framed as an emotional safety net.
Yeah, just to help people feel better.
Right. But this implies something so much deeper. Postpartum stress, whether that's from a lack of resources, a lack of community support, returning to work too early, or trauma, it doesn't just make a mother feel bad emotionally in the moment.
No, it is a physical.
It literally physically blocks the long-term biological upgrade of her brain. It stops the hardware from downloading the new operating system.
It absolutely redefines the necessity of postpartum care. We tend to view stress as purely a psychological hurdle to just, get over. But this biological evidence proves that chronic stress is a physical barrier.
It prevents the brain from achieving its full plasticity.
Exactly. It leaves the mother's brain stuck halfway through a critical transition.
So recapping this crucial point for you, there is a critical postpartum window for brain remodeling. And chronic stress during this window halts the dopamine-driven neuroadaptations. Knowing that stress physically halts this brain upgrade, how does this change the way you view postpartum support? It's not a luxury, it's a biological requirement.
Absolutely.
But, as clear as that is, there's a massive biological mystery hiding in all of this.
Oh, the epigenetic piece.
Yes.
Yeah.
Because we know dopamine is elevated and we know stress can stop it. But dopamine is just a neurotransmitter. It's a liquid messenger that floats between the synapses of neurons to pass along signals.
Right.
So how on earth does a temporary surge in a liquid messenger during the postpartum period manage to lock in structural gene changes that last for years
Right.
or even decades?
It is the million-dollar question. How does the brain save the document so that the changes persist long after the initial experience is over?
Right.
Well, the researchers chased this down and discovered a mechanism so mind-blowing it almost borders on science fiction. It is an epigenetic bookmark called H3 dopaminylation.
Okay, H3 dopaminylation. I'm going to need you to walk me through this one carefully, because the idea of a neurotransmitter acting as an epigenetic bookmark sounds crazy.
It does, but it makes beautiful sense when you visualize how DNA works inside our cells. So our DNA is incredibly long. If you took the DNA from just one microscopic cell and stretched it out, it would be about 2 meters long.
That is insane.
Yeah. So to fit inside the tiny nucleus of a cell, it has to be tightly, efficiently wound up around these protein spools. These spools are called histones.
Like thread wrapped around a wooden spool.
Precisely. And the most common of these spools in our cells is called histone H3. Now, normally our genes are turned on or off when little chemical tags like methyl or acetyl groups attach to the tails of these histones.
Okay, so tags on the spools.
Right. These tags tell the spool to unwind a bit so the genetic code can be read, or to wind up tighter to hide the gene. But this study found something entirely unexpected. Dopamine itself isn't just floating between brain cells acting as a messenger.
What is it doing?
It is physically entering the nucleus of the cell and attaching directly to the DNA packaging.
Okay, here's where it gets really incredible. It is acting like highlighter ink.
Yes.
An enzyme inside the cell called tissue transglutaminase 2, or TG2, it physically grabs the dopamine molecule and staples it onto the histone H3 spool.
At a very specific spot called the glutamine 5 residue.
Right, the glutamine 5 residue. It literally uses the dopamine molecule as a physical chemical bookmark to tag the DNA instruction manual, telling the cell to keep those maternal navigation and survival genes turned on permanently.
In biochemistry, they call it the transamidation of a monoamine directly onto chromatin. But your highlighter analogy is perfect. The neurotransmitter itself becomes a structural part of the genetic architecture.
It is just wild.
It is. And what elevates this paper from a fascinating animal study to a landmark piece of human science is that they didn't just find this in mice. They looked at post-mortem human brain tissue.
Yes, they look at a human brain region called the dorsal subiculum, which is a key human equivalent to the mouse's DHF.
Yeah.
And they compared women who had given birth parous women with nulliparous women who hadn't.
And they found the exact same conserved patterns of H3 dopaminylation and gene transcription. The identical biological bookmarking mechanism exists in human mothers.
The human brain uses the dopamine generated by the experience of motherhood to permanently rewrite its own genetic expression.
It really is profound.
It gives me chills. So to summarize this mind-bending section for you, dopamine acts epigenetically, binding directly to histones, specifically H3, to alter long-term gene expression. And this exact biological bookmarking occurs in both mice and human mothers.
Which is incredible validation.
So does it change how you view your own biology to know your experiences can literally attach neurotransmitters to your DNA? It entirely blurs the line between our experiences and our physical structure.
It really does. But, in science, observation is never enough. The researchers had beautifully observed this H3 dopaminylation, but they needed to prove causality.
Right, they had to prove that dopamine was actually causing the changes.
Exactly. They needed to prove that the dopamine was the actual cause of these maternal upgrades, and not just a coincidental byproduct of having pups around.
So they needed to hack the system. And to do this, they designed an experiment that sounds like it belongs in a cyberpunk novel. They used a technique called chemogenetics, specifically utilizing something called DREAD-Ds.
Yes, DREAD-Ds. It stands for designer receptors exclusively activated by designer drugs. DREADD.
It's as futuristic as the acronym implies.
It really is. So using viral vectors, the researchers went into the brains of virgin female mice that had absolutely never been pregnant or even seen a pup.
Okay.
And they specifically targeted the neural pathway that releases dopamine from the ventral tegmental area, the VTA, into our VIP region, the DHF.
So if I understand this correctly, the viral vectors basically installed artificial microscopic locks on these specific dopamine neurons, locks that don't exist anywhere else in nature.
Exactly right.
Then the researchers gave the virgin mice a designer drug, a specific chemical key that only fits those artificial locks. And when they turned the key, it suppressed the release of dopamine into the DHF.
Spot on. Now think back to our Sunny City analogy. Why suppress the dopamine? We know the maternal brain has high baseline dopamine, which causes it to downregulate or suppress its dopamine receptors.
Right.
By artificially suppressing the dopamine release in these virgins, the researchers were chemically tricking the virgin brain into mimicking the low receptor, highly sensitized state of a mother's brain.
And the outcome of that trick is staggering. Because by simply manipulating this one dopamine circuit to mimic a mother's brain, these virgin mice suddenly began exhibiting the behavioral perks of the reproductive experience.
Without ever having pups.
Right. When researchers placed pups in their arena, these virgins became vastly better and faster at pup retrieval tasks compared to normal virgins. They also showed enhanced contextual fear acquisition.
Meaning their internal GPS and environmental radar for threats had been completely upgraded.
They successfully induced the behavioral outcomes of motherhood entirely without a pregnancy, simply by mimicking the dopamine state.
And they didn't stop there. To brilliantly close the loop on the stress tests we discussed earlier, they performed one more hack.
What did they do?
In the mothers who had been subjected to the chronic maternal pup separations, stress the ones who lost their upgrade, they used viral vectors to manipulate the histone H3 mutation directly.
Oh.
By chemically rescuing that H3 dopaminylation bookmark, the highlighter ink, they completely reversed the negative effects of the stress.
They got their brain upgrade back.
The mothers got their brain upgrade back, yes.
That is unbelievable. So, to recap this final sci-fi experiment for you.
Yeah.
Suppressing dopamine in virgins artificially mimics maternal brain changes. And epigenetic manipulation can actually rescue the brain from the effects of postpartum stress.
It really highlights dopamine as the central, necessary, and sufficient switch for this specific plasticity.
So let's step back and look at what this all means. It means dopamine isn't just a player on the field, it is the master switch. We can theoretically induce mom brain just by tweaking dopamine circuits in epigenetic bookmarks. So if we can isolate the molecular switch for maternal care, what other behavioral states might just be a flipped switch away?
It's a huge question. If we synthesize this entire journey, we are looking at a complete paradigm shift. Matrescence is not a period of cognitive decline or temporary hormonal insanity.
Not at all.
It is a profound, evolutionarily conserved state of neuroplasticity. It is driven by dopamine rising, causing a downregulation of receptors, and ultimately physically altering our DNA packaging in the hippocampus.
And crucially, it's a biological process that must be fiercely protected from chronic stress if it is to succeed.
Exactly. It completely reframes how we value the work of parenting on a cellular level. It isn't draining the brain, it's building it.
It's building it. In fact, parity, the act of caring and raising children, is increasingly being recognized by neuroscientists as a key factor in long-term brain resilience and healthy aging.
Yes. This source proves that the profound, demanding experiences of our bodies are not just ethereal memories. They are indelibly written into the physical architecture of our minds.
Which brings us right back to where we started. We used to think of a diagnosis or a brain state like a clean binary X-ray. Broken or not broken.
Right.
But the diagnostic landscape of our lives is so much richer and so much more dynamic than that. The brain is a living document, constantly highlighting, erasing, and revising its own code based on what we endure and what we love.
It's constantly adapting.
We've just seen that the intense, all-consuming experience of raising offspring physically attaches dopamine to your DNA to change who you are forever. So I want to leave you with a really profound question to mull over.
It's a good one.
What other intense, non-reproductive life experiences, maybe a highly demanding career, a deep, devastating grief, or a profound, life-altering love, what else is currently in the background, quietly, chemically binding to your genome and rewriting your brain for the rest of your life?